Hello. My reason for this video is in several of my classes where I discuss the evolution of eukaryotes, we go through a certain number of details, but I have some videos that go through it in just a little more detail. And so I wanted to kind of offer like a little lecture on that, all of those extra details. Some classes might have this as an extra credit assignment, etc. So this is eukaryogenesis, the evolution of the eukaryotic cell, in just a little bit more detail than many classes might go through this. So if you were to go to the earliest rock layers where you can find fossils of living things, you don't see this. These are big complex cells. Look at the size of those cells. Look at how complex they are. Heck, I mean, you could actually see these without a microscope. These are ciliates, and a couple of them are actually measured in millimeters. A single cell. This one on the right, this might look like a worm, but that's a single cell. All right. So these are big complex cells. They've got the cilia, etc. These ciliates, they would be examples of protists. When would we find them in the fossil record? Well, not from the beginning, all right? And then just a quick review of fossil history. All animals would be known from these two yellow regions, all vertebrates just from the orange region. And eukaryotic cells are, in addition to more recently, these two yellow regions, only known from the green region. So if you were to start at our planet's beginning, say 4.5 billion years ago, for the better part of 1 billion years, there would be no life on Earth, no life during this time period. And then 3.5 billion years ago, if we go by fossils, maybe a little bit older, 3.8 billion years ago, if you go by chemical traces, there is life on Earth, fossils of life. We have a living planet, yes. But it's only bacteria, the prokaryotic cells. And only prokaryotic cells are known for 1.5 billion years, maybe more, maybe a little more. So our planet, for the longest time, had life, but it was only prokaryotic life. These small, simple cells like that, those are prokaryotic cells. Look how small they are compared to the ones you just saw here. All right. So, and these are archaea, by the way. That's kind of interesting. So there are these two kinds of prokaryotic cells. Archaea, you just saw some of those. Those are bacteria, but ones we don't think of that often. Many live in very extreme environments, although some live in our intestines as well, and some in ocean water. You don't have to be in, boiling water or acid pools near volcanoes to find archaea, or deep sea trenches, but many of them do prefer those environments. So many of these archaea live in extreme environments. And then there's the eubacteria. So they go back very early. That's another lecture, but archaea and eubacteria are both known from early in life's history. And we think that the early Earth did have many extreme environments. So as you go from, say, 3.5, maybe even 3.8 billion years ago to 2 billion years ago, there are two kinds of living things, two domains of life: eubacteria and archaea, for 1.5 billion years, at least a third of the history of life. This is what life looks like. But if you were to come back 1.5 billion years ago, there would be three. You would have eubacterial cells, you would have archaeal cells, but now you would have more complex eukaryotic cells. So if we were to discuss the history of life, obviously, where did these guys come from? All right, that's significant in the history of life, but also because not only do these compose ciliates or green algae or amoeba, etc. This is what composes the fungi, the plants, and the animals. So animals, plants, and fungi are multicellular kingdoms of eukaryotes, made of eukaryotic cells. So where did the eukaryotic cell come from? That's obviously then important to us because we are composed of these eukaryotic cells. So these tiny little things are the prokaryotic bacterial cells. Somehow they get to be eukaryotes. How does that happen? Well, This is going to be an overview, rapid, enough. But I've kind of stacked the conversation just a little bit in that prokaryotic cells do vary. The typical ones are, say, maybe a micron wide. So the one that causes Lyme, the one that causes Streptococcus, the E. coli living in your intestines, 1 to 2 microns long. A micron is a millionth of a meter or a thousandth of a millimeter. However, this bacterium, it's almost a millimeter. All right, this one's almost a millimeter. They're 3/4 of a millimeter. So my point is, while I'm talking about bacteria and saying, prokaryotic cells, small and simple, eukaryotic cells, much bigger, much more complex, that's not entirely true. There's enormous variation among microbes. And so some bacteria are actually larger than many eukaryotic cells. Some bacteria can actually be seen without a microscope. This bacteria, and the cyanobacteria, which I'm going to mention, has a lot of internal membranes. All right, so the cell membrane folds in a lot. So, eukaryotic cells, lots of internal membranes, bacteria, no. But some bacteria do. So it just, once again, we want to just say general things about bacteria to help understand them. But there are so many diverse bacteria that, there are exceptions to almost every rule. All right. So somewhere between 2 billion years ago and 1.5 billion years ago, from those bacterial cells, we get more complex eukaryotic cells, which compose the protists, the animals, algae, plants, and fungi. So how does that happen? A big part of it, and the primary focus of this conversation, is cells living inside other cells. All right, so cells living inside other cells play an enormous role in eukaryogenesis, the genesis of the first eukaryotic cells. So let's imagine that this is a bacteria, that part of this process would be, now a bacteria lives inside the larger bacteria. If they were to just help each other out, we would refer to that as a symbiosis. But the fact that one of them lives inside the other is called an endosymbiosis. Okay. Now, what's the evidence for this idea? I mean, it's great to have ideas. What's the evidence? Well, if you were to take genes, let's take like subunits of RNA, subunits of ribosomes, the rRNA sequences. These are things that all living things have in order to make proteins, ribosomes. And if you were to start comparing sequences, we would then get a family tree. The eubacteria, they seem to be related to each other here in purple, forming a family tree with various branches that their sequences seem to derive from a common ancestral sequence. The archaea seem to derive from a sequence. And if you look at all eukaryotic sequences, it doesn't appear out of nowhere. So eukaryotes didn't magically appear. They seem to have come from one of those pre-existing cells, and specifically the archaea. All right, so the primitive archaea formed a lineage which would lead to the eukaryotes. So lots of genes point to this pattern. Now there's another pattern, but I'll be getting to that when we talk about mitochondria. So many genes, if you compare the 3 living things, the 3 domains which were evident after 1.5 billion years ago, maybe a little earlier, the eubacteria, the eukaryotes, and the archaea, eukaryotes and archaea cluster together. They seem more closely related. And so eukaryotes seem to be related to those archaea. Since the archaea come first, a billion years first, it seems that archaea gave rise to the host cell of the eukaryotes. All right. Now, one of the problems with archaea is they're very diverse, and a lot of them live in very extreme environments that we don't get to. All right, we're not actively sampling, sediments a mile underneath the bottom of the ocean, and certainly not extensively. And once we do, it's not like we're collecting cultures of bacteria there, because a lot of them require like high pressures, high temperatures. It would be very hard to raise them in a lab. And so what we often get is evidence from DNA. DNA samples are then amplified, just like you could take a small amount of evidence of DNA from a crime scene and make enough to test. You can get, environmental samples where a little bit of DNA is amplified. And you can then identify the existence of microbes that you've never seen. You have their DNA, so this is proof that they exist from these hard to reach areas. So not only are archaea known to be the closer relatives of eukaryotes, there is a recently discovered group of archaea called a superphylum. All right, it's called the Asgard superphylum. So the Asgard Archaea. The reason for Asgard is because they were originally found in a deep water trench in the Arctic known as Loki's Trench. And then a lot of them, of the bacterial sequences, they were given names once they were found, and it was called Loki Archaeota or Odin Archaeota, or named for various Norse gods. And so Asgard Archaeota is now the accepted one. And so as you look at these new archaea, which had not previously been understood. Once again, we're searching for new kinds of archaea. We don't know all of the ones that exist, especially the ones which are in hard to reach places. And very recently, some have actually been cultured. All right, so that actually it's not just DNA sequences now, it's actually cultured. They are more similar to eukaryotes than any other known archaea. So out of all of the archaea, this group seems to be the sister group of the eukaryotes. No other archaeon yet discovered is closer to eukaryotes than these. And they have genes for the cytoskeleton more closely related to those of eukaryotes. That's important because one of the things eukaryotes can do is things like phagocytosis. They can move like an amoeba. No bacteria can do that. But these bacteria have more of the genes that let eukaryotes do it than any other prokaryotic cell. They can produce membrane vesicles. They can have protrusions from the membrane, so things can stick out from the membrane. And the ones cultured have been observed that this Asgard archaeon can, kind of be next to other types of bacteria. The paper I had read, 2 different kinds of bacteria. Protrusions can kind of contact it, and they live in a symbiosis where this eubacterium is giving the Asgard archaeon something that it wants, and vice versa. That becomes very important in our next step. And so this archaeon, which is more similar to eukaryotes than any other archaeon which is known, has a more flexible cytoskeleton. It can form vesicles, and it can form symbioses with other microbes, all kind of really important for what I'm about to go through here. I'm just going to make this a little bigger. Sometimes I regret doing that, just, this computer setup. So they've been observed here. Now, one of the things we don't know is at some point the nucleus comes about. And I'll be honest, I've tried to read as much as I can. There are a couple of models for how this might have occurred, and I'll present 3 different ones here. The first one is a nucleus really that big a deal? Well, so anyway, yes, no, but here's one of the reasons I asked the question. Some bacteria are known to wrap their DNA in a membrane. Let's not call it a nuclear membrane because, it might not be homologous and we'd have to talk about pores, etc. But if the nucleus is, all of the DNA is in one region wrapped in a membrane, some bacteria do that. Remember I said bacteria vary? Right, so perhaps a nucleus isn't that big a deal for a bacterium. Wrapping DNA in a membrane, it has been done, okay? There are bacteria which have the same proteins which form the nuclear pores that are found in eukaryotes. Once again, I don't know if these are connected, that a bacterium that can wrap its DNA as a membrane, if that has any relation to the evolution of the nuclear membrane. I don't, that isn't known. However, it is possible that an Asgard bacterium, somewhere between 2 billion years ago and 1.5 billion years ago, already had a nucleus, okay? A second option, there are some evidence, and some have proposed that a virus was involved. All right. Now, there's a couple of reasons for saying this, and viruses, are fascinating if we want to talk about the origin of life, a whole nother set of hypotheses there. But here, let's look at this. Here's a normal bacteria. One of the things that makes bacteria different from eukaryotes is that with bacteria, you have the DNA in orange there. That is transcribed into RNA. All right, so transcription occurs here. And then while transcription is going on, ribosomes can attach to the RNA and do translation to make proteins. In other words, the DNA is sitting there, all right, and transcription and translation can occur at the same spot. That is not the case in eukaryotes, because transcription occurs in the nucleus where the DNA is. The RNA leaves, and then translation occurs in the cytoplasm. So in eukaryotes, there is a separation between transcription and translation that we do not see in bacteria. Okay, so it has been observed that there are viruses which can infect a bacteria. And the bacterium, which is infected by a virus, now partitions its insides so that transcription is occurring over here, say on the viral message, and that is physically separated from the area where translation occurs. So if eukaryotes, the function of the nuclear membrane is to separate transcription occurring with the DNA in the nucleus separate from the translation occurring in the cytoplasm. Some viruses kind of achieve that, all right? And so maybe the infection of a bacterial cell with a virus had a role in this partitioning, which would have a role in the eukaryotic cells. Viral infections, in this case here, also uses a GTP cap on the RNA transcript, something eukaryotes do as well. All right, so if you were to make this hypothesis, a couple of lines of evidence would support that. Okay, so if the birth of the eukaryotic cell was a cell living inside another cell, there was a host. All right, and once again, the host seems to have been an archaean. That's one of the two domains of prokaryotic cells, and specifically one of these Asgard Archaeans. If we compare to the Asgard Archaeans alive today, they are thought to have been anaerobic. Right, the first half of Earth's history didn't have much oxygen. Eukaryotic cells were born right around the time oxygen was starting to become in the atmosphere. It metabolized hydrogen gas, very important because that symbiosis I had mentioned a minute ago and the one that I'm about to mention, it's possible that an archaean, which needed hydrogen gas, developed a symbiotic relationship with a eubacterium which produced hydrogen gas, and that's how they lived together. Once again, these archaean already have features of the cytoskeleton, which was similar to eukaryotes, and we're not quite sure where a nucleus came from, but perhaps a nucleus as well. So that's what we think the host here in tan was about. But if a cell is going to live inside another cell, what about the symbiont? Can we say anything about the cell that would end up then inside? All right. And I'm sorry, here is that description of the symbiosis here. There is this, in 2020, it was formed that this Asgard Archaean can develop these protrusions and can form symbioses with two different eubacterial cells. So I just represent those here. Okay. So it has long been known that a group of bacteria known as the proteobacteria, these would be eubacteria. So remember, there's archaea, that's one domain, eubacteria, that's another domain. The eubacteria include the proteobacteria, and they look like the mitochondria of eukaryotes, which I'm about to talk about. They have about the same size and shape. They divide by binary fission, the same way that bacteria do. So mitochondria, they look and act like bacteria. They have a circular chromosome, just like bacteria do. When you look at the genes and the gene sequences in these genes in mitochondria, they look like those of bacteria. They are sensitive to the same antibiotics. So antibiotics which harm bacteria, harm mitochondria. These are using DNA codon choices, which are like bacteria, not like eukaryotes and the nucleus. They start translation differently, like bacteria. They even have things like plasmids. So it's long been known that these mitochondria, an essential part of the eukaryotic cell, that it looks an awful lot like bacteria living inside the eukaryotic cell. Okay, so as we look at eukaryogenesis, one of the founding events, perhaps the founding event, perhaps what started all off, is a proteobacterium adapted to live inside a larger eubacterium. I'm sorry, the Asgard archaeal host. Is that possible? Well, a hard part is it doesn't happen often. All right, bacteria, I mean, they have cell walls, they don't tend to be very flexible. So bacteria living inside other bacteria, not incredibly common. It can happen because, I mean, some bacteria, they try to do that. I mean, they're predators. They invade other cells, you know, and with the idea that they're going to use that then as prey. And so some bacteria do invade other, you know, cells. That's their job. All right, and so so bacteria can certainly enter the cells of other bacteria. And if you look in, there's an insect known as mealybugs. If you look in their abdomens, there's a eukaryotic cell known as a bacteriocyte. Here's the bacteriocyte. But in the bacteriocyte, there are bacteria that have smaller bacteria living inside them in an endosymbiosis. So here, there are small endosymbiotic bacteria living inside larger bacteria, which then live inside a larger cell. And so the model of a bacteria, an archaeon, which then had a eubacteria living inside of it, which would be the ancestor of mitochondria. Here we see an endosymbiosis with bacteria living inside other bacteria. And I'd read that there was another example of this, which is currently known. So this is then possible. Now, if you were to say, all right, well, of the proteobacteria, are there any modern members, you know, which seem especially closely related to mitochondria? There have been different papers making, you know, different arguments here, but there seems to be some evidence for a group known as Rickettsia. They include in our modern world. So once again, a modern member of this group, the Rickettsiales, is not necessarily, you know, identical to, or, you know, whatever ancestral form might have entered cells. But this modern members of this bacterial group, they include endosymbionts and obligate intracellular parasites. So the proteobacteria, which seem most closely related to mitochondria, today, most of the group live inside other cells, either as endosymbionts or intracellular parasites. So 2 billion years ago, then the question is, did an ancestral form of this already have some adaptations for this? So if you have an Asgard bacteria that wants to have a partner and a proteobacteria that, you know, has some adaptations for being inside another cell, is this then how all of that began? Now, before I come back to that, just one third idea, and this gets us back to the origin of a nucleus. If we're talking about cells living inside other cells, some have proposed, well, could we add one more cell to the mix? So let's say you have an Asgard archaeon in an endosymbiosis with this eubacterium, okay? But this one hasn't entered that cell, so they're just symbionts. If you have a third cell, which is a bit flexible, that ingests both of them. If that were the case, if this pink cell, a third cell, ingests both of them, might this then be the origin of both the mitochondria and now the nucleus? Might the Asgard Archaea and its cell membrane just become ultimately a nuclear membrane, and maybe the genes of this original thing are lost, entirely or to some degree, and so that what becomes the nucleus was just the DNA of the archaea? Another idea. But anyway, let's get back to, so then the archaea would become the nucleus, the eubacteria, the mitochondrion, and the third cell then would contribute then the cytoplasm in this model. Right. So if this seems to you that, this is kind of unusual, or like, what are the odds of this? That's fine. But once again, the claim is not that this is easy or, like expected, because it is thought that the eukaryotic cells arose after a planet full of bacteria had had 1.5 billion years pass. A third of life's history passed without eukaryotic cells. So this is not easy. All right, this isn't commonplace. This would have been an extremely rare event. And all eukaryotes seem to come from this one ancestor. All right, so this occurred once after, at least a third of the history of life, and nothing similar to make a eukaryotic type cell has happened since there have been endosymbioses, but not to make something eukaryotic-like. So this is unlikely. This would not have been a commonplace event. And I say that, and I'm kind of a little torn. Well, because we haven't sampled our planet very much. I mean, the bottom of the ocean is not very well understood. Microbes have been found from 7 miles below the surface of the ocean. All right, as far down as we can go, there are microbes. It is thought that maybe, I don't know, upwards towards 40% of 40% of prokaryotic cells are actually these subsurface microbes. So my point is, I don't want to say how likely might this be, because we have not sampled much of the prokaryotic diversity that our planet has. And especially if we're talking about Archaea as being one of the 2 partners, they are most common in these extreme environments. So if we aren't really sampling extreme environments where the prokaryotes most closely related to eukaryotes would be found, then there's a lot that we don't know. Okay. One quick thing just here, if you were to weigh all of the biomass on Earth, viruses would weigh this much, animals would weigh this much, protists would weigh this much. Look at the archaea. There's a lot of those. All right, the weight of the archaea on Earth by far outweigh the animals. All right, there's fungi. And now if you look at the eubacteria, even more. Our planet has an awful lot of archaea, far more than all the animals by weight, and far more eubacteria. And so there's a lot that we don't know about these. There's a lot of kinds which haven't been sampled because of where they live. And so all of the complex interactions which might occur between them, these are things that we don't know. And plants are even more, but we'll get back to that. All right. So, the nucleus is a hard thing to pin down. There are a couple of ways that a nucleus might have arisen, and as far as from my humble reading of the literature, I'm not sure if any of the paths seems to be more definite than another. But this endosymbiosis, where a eubacterium living inside an archaean, and this eubacterium being the ancestor of the mitochondrion, this seems to be one of the first events in eukaryogenesis. Maybe the first event. Maybe this is what started off. If you were to pinpoint, these are the cells that were the first ancestors of eukaryotes. It might have been the first ones to get a eubacterium inside an archaea. All right, so let's say that that's here. The original eukaryote following the endosymbiosis of a eubacterial ancestor of mitochondria. Maybe it had a nucleus, hard to say. Maybe the nucleus comes later. Well, if you were to look at all of the eukaryotes alive today, okay, they are, say, here in this family tree of Protists, and here animals would be there, fungi would be there, and land plants would be there. So lots of different kinds of Protists, plus then 3 multicellular kingdoms of animals, plants, and fungi. We use a term, LECA, to be the last eukaryote common ancestor. So what is the most recent common ancestor which would share ancestry with, the animal lineage, and then maybe the most primitive lineages? Now, biologists have long argued about the branching of the Protist family tree, including which ones are the oldest, but things like Giardia seem to be near the base. So I like that, because that's, I think consistently been shown to be one of the most primitive eukaryotes, if not the most primitive. I've got pictures of eukarya, which obviously make my videos better the more pictures I have. And while I was in the Peace Corps, I had Giardia for 2 years. And so, this thing living inside me, we build up a relationship. So if it's okay with you, I'll just be going with the consensus and using Giardia as the example of the most primitive lineage of Protists. So what is the most primitive lineage? My fault. So if you ask, what's the most primitive ancestor which gave rise to both Giardia and animals, the most recent one, I'm sorry, let's call that LECA. Well, LECA is not this thing that just had the symbiont ended inside. So there would have been a road from this one to this one, all right? It's not an instantaneous thing to start branching and forming the lineage to Giardia. And there would have been changes along this pathway, giving rise to more branches of the family tree. Maybe they've died in the past 1.5 billion years. Maybe we have yet to discover them. But certainly, if we look at what eukaryotes all share, if there are things that eukaryotes share that no bacteria has, then some of this would have arisen in that time period, in between that endosymbiosis and LECA. So when we look at things, just asking what do eukaryotes share? Obviously, they share a nucleus. They have a mitochondria. They have an extensive membrane system, which use Golgi and an endoplasmic reticulum. If you look at the way amoeba move, clearly the cytoskeleton made of actin, which can be reorganized, is important. They have true cilia and flagella. Now, bacteria do have flagella, but there's different kinds. The flagella in eubacteria is different from the flagella in archaea, and that's different from the cilia and flagella in eukaryotes. So there's a whole bunch of things. At some point, eukaryotes get a new kind of flagella. At some point, eukaryotes undergo mitosis as a way of separating the DNA material in cell division, something that no bacteria does, et cetera. So once again, somewhere between the first endosymbiosis and LECA, all right, we think that, a whole host of things evolved. And once again, this could have been, tens of millions of years, hundreds of millions of years before the lineage, which would lead to the modern Protists like Giardia would have come from. So there's all kinds of genes as well. Okay. Now, specifically, I'd like to address how would this eubacterium have to change before it becomes the ancestor of the mitochondria, which we see today. All right, so if a cell lives inside another cell, if that's a eubacterium living in a larger cell, that's not a mitochondrion, that's the ancestor of the mitochondria. So how does this thing have to change before it becomes this thing in LECA? Well, one of the things that both mitochondria and chloroplasts have is protein transporters, which allow things to be made in the cytoplasm to get pumped into the symbiont, which are needed, and then things from the symbiont to be pumped into the cytoplasm. So if this is an endosymbiosis, both are benefiting from each other. And then these transporters are what allows that. We can transport stuff back and forth so that each of these partners can get what it needs. All right. A bacterium tends to have lots and lots of genes. Mitochondria, not so many. And so there has been a great reduction in the number of genes by the time we get to mitochondria. And then later, the same could be said of chloroplasts. There are far fewer genes, so a reduction of these genes. These two might not have worked together, that closely, but these two do. All right, so the mitochondria is tied to the biochemistry and the program of the larger cell. So when the cell is dividing, the mitochondria are involved and coordinated with that. The cytoskeleton interacts with mitochondria. The membrane system interacts with mitochondria. And so this would have needed to change. Also, if you look at mitochondria today, there's an outer membrane and an inner membrane. And for all of those biology students who have studied cellular respiration, this is where the electron transport system is. The hydrogen ions go out here, the electrons go in there. And when the two come together, this is when you make ATP. All right. So the symbiont would then have to change its inner membrane as well. Somewhere I point out, somewhere. Did I miss that in the last video? I might have. That eubacteria, the gram-negative ones, and these are the ones in the proteobacteria group, they already have two membranes. There's an outer membrane and an inner membrane. And so if a eubacteria ended up inside a larger cell, it already would have had an outer membrane and an inner membrane. And so in the mitochondria, this inner membrane has been highly modified. Now, I had mentioned that a lot of genes are lost. Mitochondria have far fewer genes than what the original the original prokaryotic symbiont would have had. But interestingly, a lot of these genes have been taken up by the nucleus. So if you were to look at nuclear DNA today, say in me, a lot of these genes are more similar to archaea than they are to eubacteria. And that makes sense because this original host cell was thought to be an archaean. But for some of the genes, it's the opposite. Some of the genes in my DNA are more similar to eubacterial genes than they are to archaea. And what was thought is that while mitochondria today only have a small fraction of the original repertoire of genes, that many of them ended up in the nucleus. So that if somewhere along the way, one of these cells died, and now, in its debris, there's DNA in the cytoplasm, the nucleus can take it up and incorporate it into its own genome. All right, so that is one of the things which can occur, that DNA can be taken up and incorporated in the genome, as can RNA. All right. So, this is the year of coronavirus as I'm making this video. So obviously, there have been DNA and RNA vaccines, which the purpose of the vaccine is that the cells are taking up foreign DNA and RNA and then, starting to make proteins from them and even incorporating them into the genome. And so DNA can be taken up, as can RNA. So if this bacteria has a DNA chromosome that it transcribes into RNA. After it bursts, the RNA could then end up in the incorporate into the nucleus as well. Once that happens, then it is a waste of energy to have two copies of this in both the nucleus and the symbiont. And so if the symbiont has mutations, which will then make its chromosome smaller, that's actually more efficient now because the genes have migrated into here. And so what has been observed, and we can still continue to observe this is easily documented, that symbionts often reduce their genomes, and many of the genes which were originally present in the symbiont end up in the mitochondria. Today, the human mitochondrium has its own genome. It still retains a chromosome of around 16,000 base pairs, which has a number of genes, most of which are for translation. All right, 13 proteins, some tRNAs, some rRNAs. So the translation which occurs inside the mitochondria, a little bit different from that which occurs elsewhere throughout the cell. We do have a lot of proteins which connect mitochondria with neighboring membranes. So endoplasmic reticulum has specialized areas called MAMs, mitochondrial associated membranes, where there are special pores for stuff to go back and forth. All right. Now, just very quickly, mitochondria do so many things. I know we associate it primarily with making ATP for aerobic respiration, and that's obviously very important. But mitochondria do other things as well. They're involved in metabolism. There's lots of things that cells need that mitochondria make. They're making metabolites. They're helping to control transcription in the nucleus. They make things which affect transcription. They help to kill cells which have started to take the steps towards cancer. And so mitochondria have lots of roles. They interact with the cytoskeleton and they can move. So mitochondria are actually quite complex, and so a full description of this topic would probably get into more of that, but I do that in other videos, not in this one. So then the question is, what kind of mitochondria existed here? Say if we were to look at, say, Giardia, the most primitive protist branch. So as we go from a bacterial symbiont to here, what changes occurred? And that's difficult because Giardia don't have mitochondria per se. So they do have a mitochondria like I have, they don't. So some originally thought, well, maybe then that's proof that the endosymbiosis actually occurred after this point, that Giardia are so primitive, they actually predate the endosymbiosis of these bacteria. But the answer is a little more, that's not true, and the answer is complicated. So Giardia, they're complicated. Do they use oxygen? They do, but they live in environments like intestines where there's not a whole lot of oxygen. So why is that? Is it because they're so primitive, their lineage dates back to a time where there wasn't much oxygen in the air, that there was a little but not a lot? Maybe, or maybe this is a secondary adaptation to a parasitic lifestyle. They don't have mitochondria, but as I'll go into the next video, they have what's called a mitosome. So so it seems to be they come after the endosymbiosis, but might they come before the symbiont became a mitochondrion? So if there's a path between the original endosymbiosis and LECA, do Giardia date to a point before there was a true mitochondrion? So Giardia are confusing. They don't have true Golgi, but they have something that looks like a little like Golgi. They don't have sex, but they do have some genes for sex. So, are they representing the primitive condition of the first symbionts, or have they changed as they were adapting to live inside the environment of, say, intestines and the like? So, so Giardia are tough in that way, but but certainly they seem to be primitive in this sense. Okay, so. So the original symbiont probably possessed maybe 3,000, 4,000 genes. But now there's probably only a handful. I'll go through some variations in just a second. And eukaryotic nuclei, including those of humans, include about 800 genes which seem to have occurred from these symbionts. So if this dies and some of the DNA is taken up, so out of, say, the 23,000 genes that the human genome has, maybe 800 of them seem to have resulted not from the Asgard Archaean, or just normal gene duplication and the like, but rather from the eubacterial symbiont. All right, and so the original eukaryote is a chimera, having genes from two different different organisms. Okay. Interestingly, while some of the genes that the nucleus has, which came from the eubacterium, some of them make proteins which get transported back to the eubacterium, to the mitochondrion, but some of them don't. A lot of them just have other roles within the cell that aren't directly linked to this mitochondrion, so that is of interest. Okay, so most of the genes which function in the mitochondria are not those few encoded by the mitochondrial chromosome, but rather encoded by nuclear genes and imported to the mitochondrion from the cytoplasm. Simple mitochondria found in protists may require several hundred proteins. Those of vertebrates, several thousand proteins. And once again, there's only a handful of proteins encoded by this mitochondrial DNA. Most of the proteins you need to make a mitochondrion function are encoded by the nucleus, made in the cytoplasm, and then transported here. Eukaryotic mitochondria typically have only several dozen genes, mostly for translation and electron transport. So if you were to ask, in mitochondria today, what's the biggest number of genes? What's the smallest number of genes? There are some mitochondria in some protists, like the branch that includes malaria, which encodes only 2 or 3 proteins, where there's another one which encodes 66 proteins. That's the biggest number of proteins encoded by a mitochondrial chromosome. So there's variations in mitochondria today. Not all mitochondria are the same. So this lineage has some which vary from others. Relatives of Euglena, all right, they have genes in their mitochondria that my mitochondria don't have, and those genes are homologous to those of proteobacteria. So once again, increasing, the evidence for that endosymbiosis. Typically only 16,000 to 18,000 base pairs. And this can vary from lineage to lineage. So, here's a nematode with a smaller number here. Plant mitochondria can be a bit bigger. All right. They can have hundreds of thousands of nucleotides. Once again, mine only has 16,000. One even has 2 million nucleotides. So mitochondria came from the early eukaryotes, and they have changed over time. Mammals, for example, share the same genes in the same gene order. All right, and so, plant mitochondria are different from animal mitochondria. They differ in their genes, the gene order, the amount of the DNA which is coding for a gene versus not. So animal mitochondria are more efficient than those of plants, more than 90% coding. There can be variations in how many copies of mitochondrial chromosomes there are. Plants have more copies of mitochondrial chromosomes per cell. There can be changes in which codons get used. And so AUA can mean different things in different mitochondria. Okay. And then finally, what do mitochondria look like? Well, we are familiar with aerobic mitochondria. Most eukaryotes, the mitochondria are breaking down sugar using oxygen to produce 36 molecules of ATP. There are variations, however. There are anaerobic mitochondria, which you can find in some invertebrates like worms and mollusks, which live where there's not a lot of oxygen. So, for us, mitochondria, they, use oxygen to break down sugar, produce a lot of ATP, but not all of them do. Some don't use oxygen ever, and they generate a smaller number of ATP molecules per glucose molecule. And then there are things called hydrogenosomes, which seem to be descended from this original proto-mitochondrion. They don't use oxygen, and they generate a little ATP, but they ferment it to produce hydrogen gas. All right, and remember that endosymbiosis with an archaea that I mentioned, the production of hydrogen gas, that is what modern archaea can use in a symbiosis. However, there's even simpler ones, these tiny little things, all right, called mitosomes, which are small. They're much smaller, and these can be in things like Giardia. Once again, Giardia may represent the most primitive eukaryote alive today. It doesn't produce ATP at all. It's just forming clusters of iron and sulfur, which are useful. Both of these lack a chromosome. So mitochondria, these and these, possess chromosomes which have been inherited from the proto-mitochondrion, which was a bacteria. But the gene number got smaller and smaller. These hydrogenosomes and mitosomes, they are now completely without chromosomes. So getting back to that LECA family tree thing, which of these happened where? Did the proto-mitochondrion become this type of mitochondria and then later got modified into this one, and this one? Or do Giardia, does it branch from the family tree here so that this developed into a mitosome in Giardia and this developed into a hydrogenosome in some protists, and that the changes which would make this into a true mitochondria, do they come later? All right. So that's a question. Forgive me. I just want to make sure that I am not in a room that someone else needs to be in. So I'm going to just pause the recording a second. Oh, no, sorry, paused the wrong thing. Okay. So the mitochondria evolved from a eubacterium living inside a larger cell. And this is one of the first events in the generation of the eukaryotic cell. But we are not done with endosymbiosis in the process of eukaryogenesis. So I'd now like to talk about the chloroplast. Chloroplasts evolved from a type of bacteria known as cyanobacteria. They're often referred to as the blue-green algae, but they're not algae. They're bacteria, and they're very diverse. They represent a kingdom of bacteria. Now, bacteria can produce different photosynthesis through different ways. So there's different kinds of photosynthetic bacteria. So there are the purple photosynthetic bacteria, but cyanobacteria, they are different. They're green because they have chlorophyll. The chlorophyll is located on internal membranes, which form these structures known as thylakoids. They take carbon dioxide from the air to make their carbon, and they release oxygen gas as a waste. So there are different types of photosynthesis in bacteria. The cyanobacteria they're different. And if you're saying, well, that isn't very exciting because don't plants do that? Aren't plants green? Don't plants have thylakoids? Don't, plants have, take carbon dioxide from the air and make it into sugar? Don't plants release oxygen gas? They do, all right, because plants evolved from green algae, and green algae are green because they have cyanobacteria living inside them. So cyanobacteria are really important. First off, they come first. All right, so cyanobacteria are known in the fossil record from before 2 billion years ago. All right, and so maybe 2.2, 2.3 billion years ago. And if you were to look at Earth's history, you could divide it in half. For half of Earth's history, there isn't any appreciable oxygen in the Earth's atmosphere. All right, but then you get to around the point where oxygen, when cyanobacteria appear in the fossil record, and now oxygen levels begin to accumulate, not as high as they are today for a long time. So I mentioned, once again, did Giardia come from a primitive time period where oxygen had begun to develop? So early in this blue block, there would have been oxygen in the air, but nowhere near as much as there is today. Did the early eukaryotes evolve with a little bit of oxygen, and do Giardia, come from that time period? Okay, well, in any case, cyanobacteria, they are one of the most important living things in the history of our planet. They changed the planet. All right, they took a planet that didn't have appreciable oxygen in the atmosphere, and following them, there is appreciable oxygen in the atmosphere. So one of the most important events in the history of life. Shortly after that, the first eukaryotic cells and protists evolve. So protists evolve in a world that has oxygen. Not as much as modern times at first, but nevertheless, these cyanobacteria, they helped set the stage for life on Earth as we know most of it. Clearly, there are still bacteria that live in anaerobic environments. So cyanobacteria, they are gram-negative bacteria. They have two membranes, an inner cell membrane. This would be the same as the plasma membrane. They have an outer membrane. They have a circular chromosome. They have an outer membrane and inner membrane. Between the two membranes, there's a cell wall made of peptidoglycan. This is what bacterial cell walls are made out of. And their inner membrane folds inward. I know we associate internal membrane-bound structures with eukaryotes, but some prokaryotes also have internal membrane-bound structures like thylakoids here. So here you can see a gram-positive bacteria, which has one membrane and then a cell wall. Gram-negative bacteria have a cell membrane, a peptidoglycan cell wall, and an outer membrane. So in mitochondria, this would then become the outer membrane, the inner membrane, and the same would be true of chloroplasts, which have an inner membrane and outer membrane. There are some chloroplasts which actually still retain peptidoglycan in the cell wall, but most would lose that, and that would just become then a space. All right. And so cyanobacteria, the blue-green algae, they are green doing photosynthesis. They have thylakoids where the chlorophyll pigment doing the light harvesting is found. Okay, and clearly they're important. One of the reasons they're important is they're producers, right? Just like plants are producers making sugar, and lots of things eat plants. Lots of things eat cyanobacteria. I mean, for example, sponges do. All right, they're filter feeders. Next, you're going to see a rotifer, which is an animal which filter feeds and thus gets. Oh, I'm sorry, that's not a rotifer. Rotifers. I'm sorry. Rotifers are animals which filter feed as well. But then there can be microbes which eat a cyanobacteria. So look at this paramecium. See how green it is? Why is it green? Because it's eating cyanobacteria. All right, so they use it for food. So just as lots of animals eat grass, lots of animals ingest cyanobacteria as food. Some paramecia can actually keep the cyanobacteria around for a bit with a relationship where they are having a benefit from these cyanobacteria living inside them. Now, they're not true endosymbionts. I don't think they last that long, but nevertheless, you can kind of see, kind of maybe even a temporary stage here. So lots of cells ingest cyanobacteria. We think that once upon a time, and this happened one big time, that a cyanobacterium which had been ingested escaped to the point where it now lived inside the cytoplasm of a larger cell. Not in here. So if this was the original endosymbiosis, and this is LECA, the ancestor of Giardia and animals, we think that it occurred here, all right, in this branch, which would lead to a group known as the Archeplastida. And we think that this occurred later in eukaryote history, maybe about 1 billion years ago. 1 billion years ago, we think that a cyanobacterium entered one of the early amoeba of this lineage. So this is a lineage kin to amoeba, the Archeplastida. And that from this, we would get the red algae, the green algae, and the land plants, as you will see. All right, so Archeplastida has a number of modern lineages alive today. Red algae, green algae, land plants, and then actually a couple of other lineages as well, which I'll point out in a second. So there are different types of photosynthesis in eukaryotes, but some of these have evolved from a common ancestral form. So Archeplastida include the red algae, which would have occurred after that. Now, notice here's a red alga, which is green. Okay, all of the members of this group do possess chlorophyll, and some red algae, have a greenish color. Once again, they do have chlorophyll A. That's different from the brown algae, which I'll get back to later. So here's a red alga, which is green, once again, because of the chlorophyll which it possesses. It's a member of Archeplastida. As are the green algae. Here are different green algae. They are green, all right? And from the green algae, we get land plants, all right? So there are different kinds of green algae. Some of them are more closely related to land plants than others. So here's a green alga of the group Charophyta. This green alga is more closely related to land plants than the Chlorophyta here, like Spirogyra and Chlamydomonas. So a lot of the green algae that you might know, they're here in the family tree, the Charophyta, they are more closely related to land plants, having some of the features of land plants here. Now, before we talk specifically about those chloroplasts, all right, so it would be in the Archeplastida, the red algae form a group, the green algae form a group, land plants come from that. I just want to make the point of how many plants there are. If you were to weigh all of life, if you were to weigh all of life, as I pointed out, the eubacteria outweigh, so if that's second line is the eubacteria, that outweigh the fungi, the protists, the animals, the archaea, all put together. There's a whole lot of eubacteria. But look at the plants. Look at how many plants there are. Just look at the weight of the plants on planet Earth. If you were to weigh all of plants, they would weigh more than 4 times all of the other groups combined. And I say that because cyanobacteria live inside these plant cells. Cyanobacteria descendants are chloroplasts. So not only did cyanobacteria put oxygen in the air in the first place, without those cyanobacteria, we wouldn't have the green algae. And from the green algae, we get land plants. So when you look at land plants and look at their success, in a way, you're looking at the success of the cyanobacteria because their descendants, the chloroplasts, live inside the land plants. It is those cyanobacteria which make the plants green, which allow them to do photosynthesis, which is why they can fix carbon, which is why they produce oxygen as a gas. All of these were features of the cyanobacteria, and all of these are features of the chloroplasts, which have descended from them. So cyanobacteria, clearly among the most successful organisms on the planet and the most significant in the history of the planet. So the endosymbiosis, where here at the Archeplastida, a cyanobacterium ended up inside an ancestral amoeba, clearly very significant. All right. So this gives us the algae, which we see here, clearly green. So cyanobacteria, they look like this. And if you were to look at the chloroplasts of these green algae, they are similar, all right? So they have a chromosome. They have outer and inner membranes. They have thylakoids. Some still possess peptidoglycan. I'll get back to that. And so the chloroplast is a descendant of cyanobacteria, all right? The Archeplastida include the red algae, green algae, and land plants, which I mentioned, but also 2 minor groups, the picozoans, which have a plastid but don't use it for photosynthesis, and the glaucophyte algae. There aren't many of those, but they still have peptidoglycan in their cell wall, evidence of how primitive they are. So there's only 15 species of those, but they retain peptidoglycan, once again, showing how that the chloroplast is descended from a bacterium which had peptidoglycan. These picozoans seem to have lost all trace of the chloroplast. And then you get to the red algae, the green algae, and the land plants. Okay. Now, as time went on since the endosymbiosis, these chloroplasts, I'm sorry, my fault, before I go into that. Here's a weird question. Did this cell, which started off Archeplastida, did it have chlamydia? That's an odd question to ask. But all the cells of modern Archeplastida, or the Archeplastida alive today, they seem to have 20 or 50 genes which didn't come from Archaea, didn't come from the eubacterial ancestor of mitochondria, and didn't come from cyanobacteria, but which seem to come from chlamydia. It has been hypothesized that if you ask why did this cyanobacterium escape phagocytosis? Lots of things eat cyanobacteria for food. Why did this one escape and not get digested? There is a model by which this cell took this in for food, but had an infection of this group of bacteria which infect inside cells. And that this cell then maintained itself by kind of transporting stuff from the big cell into the cyanobacteria, and the cyanobacteria to the big cell, and it benefited at first. That it was instrumental in establishing this relationship where the cyanobacterium could stably live inside the host. And eventually, the chlamydia was not needed and was lost. That 20 to 50 genes would end up in this nucleus. But then later members of this lineage would maintain a relationship between the cyanobacterium and the host cell without chlamydia, so that it was no longer needed. So that's an interesting, one more cell which might have been involved in establishing this early endosymbiosis. Okay, but so that's interesting. But getting back here, these green algae, all right, they have chloroplasts, which are similar to cyanobacteria. So if you were to compare a cyanobacterium to the chloroplast in a green alga to the chloroplast in a land plant, there are differences. These two chloroplasts don't have as much DNA. They do have circular chromosomes, but not as much DNA. At some point, the thylakoids would form stacks known as granum, or grana in plural, that would no longer be connected to the inner membrane. All right, but clearly, the overall structure between the cyanobacteria is similar. Most chloroplasts lack peptidoglycan in the cell wall, but as I mentioned, there is that one primitive group of algae, the Glaucophyta, where there is still peptidoglycan in the cell wall. Chloroplasts today need proteins, some 2,000, some 5,000. Most of these are not encoded by this chloroplast DNA. Most of them are encoded by the nuclear DNA. Chloroplast DNA typically only encodes about 100 proteins. So just as in mitochondria, proteins then are made in the cytoplasm and shipped to the chloroplast. The genome of the chloroplast, this chromosome, has gotten smaller. It has been reduced, all right, encoding some genes for photosynthesis and then a number of genes for translation. Like bacteria, the genes occur in groups known as operons. So eukaryotes don't have operons. Bacteria do, where maybe 3 genes which make proteins which all function in the same metabolic process are located in a row. Called an operon and regulated with the same regulatory mechanisms. Bacteria have operons, cyanobacteria have operons, and so too do chloroplasts, which are descended from cyanobacteria. And so there's that. And then if you were to look at chloroplasts, once again, they are descended from bacteria. So their RNA polymerase is more similar to those of bacteria compared to the, say, the algal cell or the plant cell, what's used in the cytoplasm. Their ribosomes, the way they replicate chromosomes, their membrane lipids are all more similar to cyanobacteria. All right, and that's in addition to things I've already mentioned, like antibiotic sensitivity, the circular chromosomes, et cetera. So chloroplasts are the descendants of cyanobacteria, which adapted to live inside eukaryotic cells in an endosymbiosis. All right. One of the keys that would develop over time is a set of proteins which would transport stuff to and from the chloroplast and the remainder of the cell. So there are channels known as TOC channels on the outer membrane and channels known as TIC channels on the inner membrane. And because of these channels and others, you can transport stuff back and forth, including proteins, so that proteins made in the cytoplasm can be transported to the chloroplast, which needs proteins, but which doesn't have a lot of its own genes to make proteins. Okay. Now, how likely is this? Well, here's a fascinating thing. This happened once, but it seems to have happened, something similar has happened more recently, because there is an amoeba. So here's a modern amoeba, and it has a cyanobacterium living inside it. This is not a chloroplast from that original endosymbiosis. So instead of occurring 1 billion years ago, this is thought to have occurred maybe 60 million years ago. There has already been a reduction in the genes in the endosymbiont. So maybe 3/4 of them have been reduced, and a couple of them are non-functional pseudogenes. More than 30 genes that started off in the cyanobacterium are now in the nucleus, and some of the nuclear genes produce proteins which are transported to the endosymbiont. So this is not what gave rise to the chloroplast of green algae. This is something new. Maybe only 60 million years ago, affecting one kind of amoeba. But it seems to be a very similar process as what did occur then. So eukaryotes depended on endosymbiosis to give rise to the mitochondria somewhere between 2 and 1.5 billion years ago. But around 1 billion years ago, another very important endosymbiosis occurred, which gave rise to a lineage known as the Archeplastida, which gave us a number of lineages, including the red algae, the green algae, and the land plants. The last part of this lecture is step 7, secondary endosymbiosis. So primary endosymbiosis is when a larger cell takes in a bacterium. So there is a cell living in inside another cell. And so here is the chloroplast of a green alga. That's the descendant of a cyanobacterium. So here's a bacterial cell descendant inside a larger cell. This is primary endosymbiosis. So primary endosymbiosis. There are more algae, all right, here that are different from this Archeplastida group. So we have what are called the heteroconts, which include brown algae and diatoms. We have Euglenozoans. We have others. Where did these other algae come from? Okay, well, some of these, we think that they became photosynthetic because of a secondary endosymbiosis. So imagine, here's the green alga, all right? So the first eukaryotes get the ancestor of mitochondria, they get the ancestor of chloroplasts. So both of these represent primary endosymbiosis of a bacterium living inside a larger cell. But now imagine this is the green alga or red alga. And now imagine then a larger cell ingests these. So here you have a eukaryotic cell living inside another eukaryotic cell. This would be called secondary endosymbiosis because this symbiont in turn has symbionts. If this eukaryotic cell is an endosymbiont, it in turn has endosymbionts. So this is secondary endosymbiosis. So once again, the ancestor of the mitochondria inside an Asgard Archaea, primary endosymbiosis. The green alga, a primary endosymbiosis. But a green alga or red alga living inside a larger cell, that would then be a secondary endosymbiosis here, okay? So here you see Euglena. It's a kind of protist alive today. And as you can see, they're green inside and they can do photosynthesis. They kind of act a bit like animals. They swim with a flagella, they can ingest other things, but they look like they have chloroplasts. Are these chloroplasts the result of a cyanobacterium inside, in which case it would be a primary endosymbiosis, or is it a secondary endosymbiosis? It seems to be a secondary endosymbiosis. Not all of the Euglenozoans are green. Most of them aren't. Most Euglenozoans don't do photosynthesis, like the ones which cause African sleeping sickness and Chagas disease and other diseases. So here you see a Euglenozoan which is not photosynthetic. All right, you see a lot of them here. Once again, they can cause some human diseases. So the first members of this lineage did not have an endosymbiont inside them, but later one lineage did. So here's a Euglenozoan that now ingested what we think are descendants of green algae living inside a larger cell. All right, this then gave rise to the photosynthetic Euglenozoans, which is not the majority of Euglenozoans, it's only some of them. It's a secondary endosymbiosis. And when you look at their genes, they don't just seem to be genes of cyanobacteria, they seem to be genes of green algae. All right, the chloroplasts don't just have two membranes, they can have more membranes. All right, so Euglenozoans include some which are photosynthetic. They include some ones that look like green algae, but they're not green algae. They seem to have arisen from a secondary endosymbiosis where green algae are living inside a larger eukaryotic cell. The same thing seems to be true of other protists where an early red alga ended up inside a larger eukaryotic cell. So if a red alga, which had chlorophyll and then other pigments, ended up inside a eukaryotic cell, this gave rise to heteroconts, which have this golden brown color. They're algae. They include this Cynora, that's a heterocont alga. You can notice its color. This includes the brown algae, which can form kelps, which are, can be well over 100 feet long. All right, they can be quite complex. Here you can see complex reproductive structures in a brown alga. And they also include the diatoms. All right, so Cynora, brown algae, diatoms, they are heteroconts named after the flagella which they have. But look at the color. They're this golden brown color. They're golden brown because their plastids contain fucoxanthin, all right, which they share. And their chloroplasts are wrapped in 4 membranes. They possess chlorophyll A and C, like the red algae. They also have this fucoxanthin. But those 4 membranes are from a secondary endosymbiosis. So it wasn't that a bacterium ended up inside the early heteroconts, but rather an early red alga ended up inside the early heteroconts. So heteroconts are photosynthetic and seem to have resulted from a secondary endosymbiosis. Some of the alveolates, here's another branch of the protist family tree. The same is true. So when we look at dinoflagellates, some of them have chloroplasts inside them. So if you look at a dinoflagellate, there are different kinds, but they are often golden brown in color. And they have chloroplasts inside them. These chloroplasts are not like bacteria, like cyanobacteria, but rather they seem to be a secondary endosymbiosis. An early red alga was ingested inside these cells. Now, dinoflagellates, which have chloroplasts and can do photosynthesis, they're certainly important. But they're important for another reason, because they're inside coral. Corals are animals, but if you look inside coral animals, they then have dinoflagellates inside of them, all right, which means this is a tertiary endosymbiosis. These zooxanthellae, that's what we call the dinoflagellates living inside corals, all right? This is a tertiary endosymbiosis. So it would be the ancestral red alga, an early member of the Archeplastida, which had both a mitochondrial and a cyanobacterial endosymbionts. Those were primary endosymbioses. The early dinoflagellates, which then had a red alga inside, that would have been a secondary endosymbiosis, a dinoflagellate. But then there are animal cells from corals, which then have dinoflagellates living inside them. That's a tertiary endosymbiosis. And so eukaryotes, including some animals, have clearly depended on these symbioses. As I kind of get into my last point, however, you may have noticed that the heteroconts are golden brown because of a secondary endosymbiont. The dinoflagellates are golden brown because of an endosymbiont. But here's something that's confusing. These aren't photosynthetic, nor are these. So these don't seem to be algae. And so one explanation for this distribution is that a secondary endosymbiosis occurred here, and a different second endosymbiosis occurred here. So the secondary endosymbioses occurred twice, one in the early heteroconts and one in the early dinoflagellates. Okay, and you could explain that because the ciliates and these apicomplexans, which include the organism which causes malaria, they don't have a photosynthetic thing. So here's a ciliate. They don't do a photosynthesis with a symbiont like that. However, if you look at malaria closely, the cells which cause malaria, they do have something that looks like a chloroplast. It is not for photosynthesis, but it seems to have descended from a plastid which once did. Now, this is hard to compare because the chloroplast of dinoflagellates only has a handful of genes that are used for photosynthesis. And the organism which causes malaria only has a handful of genes, but they're not used for photosynthesis, they're used for translation. So it's hard to compare them. But it seems that relatives of the malaria organisms do have a plastid which performs photosynthesis, indicating that the plastid known as the apicoplast in Plasmodium, which causes malaria, is a descendant of a photosynthetic plastid which lost its ability for photosynthesis. And so if that is true, that would indicate that if you ask why do heteroconts have a golden brown plastid, and why do dinoflagellates have a golden brown plastid? Because instead of two separate events, once at a shared ancestor between heteroconts and these alveolates, that a secondary endosymbiosis would have occurred. And the reason that modern ciliates and the organism that causes malaria don't have a photosynthetic symbiont today is that they lost it, that they lost something that was once in their common ancestry, that the secondary endosymbiosis occurred once rather than twice. So the model is, did it occur once in the heteroconts and once in the dinoflagellates, so twice in total, or did it just occur once and some members of this lineage lost it? All right, so you don't see that today, in which case we would say that these two branches are related and the name given for them is the chrome alveolates. Okay, so eukaryotes make up most of the bio stuff alive today, especially if you consider plants. And so this lecture has kind of been asking, how did eukaryotes come to be, describing the process known as what's called eukaryogenesis. So how did the eukaryotic cell come to be? And I kind of made it as a separate lecture because, as you can see, it's quite involved. And as a result, for some of my classes, this is more detailed than most of them have signed up for. I would like to end with like a silly note, because I have a song which I made kind of on this. The first song that I used using my harp. So, this song, little harp in the background, tries to, in a funny way, sum up this incredibly important process in the history of life, eukaryogenesis, the evolution of the eukaryotic cell. One of life's important anecdotes. 2 billion years without eukaryotes. 2 types of cells were all that you could see, until the day eukaryotes made free. The process and beyond, Asgard Archaea named Asgard, already more flexible than those, perhaps a nucleus, in this post, endosymbiosis had begun. Ancestor of the mitochondria. Look at the similarities one tallies with proteobacterial group Rickettsia. Transported, modified, membrane, genes will also nucleus gain, variations in mitogenomes, mitochondria, hydrogenosomes, Cyanobacteria produce nitrogen gas, and the symbionts became the Chloroplast, perhaps with help of chlamydia. Groups of Archeplastida. Transporters who would transfer all, multiple again from some gene transfer, some retain, prokaryotic features still remain. Secondary endosymbiosis occurs as well. Eukaryotic algae inside a larger cell. Euglenozoan is something you might want to give brown pigment to heteropods. And plastids sometimes go away, who's related hard to say. More endosymbiosis is true, Euglenozoans and carbon. All these events, they might seem rather strange, but life on Earth would be forever changed. Fail to mention biologists would be remiss. The importance of eukaryogenesis.